Vamos a empezar. Primero el aviso de seguridad. En caso de alguna emergencia, la evacuación del auditorio se va a hacer en dos partes. La mitad de este lado va a salir por esta puerta, la otra mitad va a salir por la puerta de allá, y hay que mantener las escaleras libres. Por favor, nadie se sienta en las escaleras. También les pido de favor que todos apaguen sus celulares o los pongan en modo silencio y que no consuman alimentos. Bueno, ahora les voy a presentar al doctor Kenneth Birnbaum, que es invitado del doctor Luis Rosas. Él es investigador titular del Departamento de Biología en el Centro de Genómica y Biología de Sistemas en la Universidad de Nueva York. Hizo su licenciatura en Biología en la Universidad de Pensilvania, la maestría en Ciencias Ambientales en la Universidad de Wisconsin-Madison y el doctorado en Biología en la Universidad de Nueva York y en el Jardín Botánico de Nueva York. Y sus líneas de investigación abarcan proyectos de ciencia básica como el estudio del desarrollo y regeneración de plantas y evolución de la raíz en plantas, pero también proyectos de ciencia aplicada como la selección de poblaciones de algas con características deseables para la producción de biocombustibles por medio de fluorescencia, y además tiene numerosas publicaciones en revistas de mucho prestigio como Cell, Nature y Science. Le damos la bienvenida.
Okay. Well, thank you for coming out this morning, and I'll tell you a little bit about our research. You can start that movie if you can click the beginning. Great, thank you. So this is a root, and I have the drill next to it because we're kind of just thinking about the root in terms of its function, and essentially it's an organic drill, right? That's collecting nutrients and water from the soil. And here you see my students devise this system where he can image the root. This is 40 slices with a confocal using a nuclear marker. Here you're seeing every 14 minutes time lapse over about a week. So we can really watch, and you really get a sense of the root as this organism that's exploring, it's normally the soil. It actually hit itself and it's wrapping around like a snake, and that's why you see it hitting the larger cells here. And so normally my lab studies a lot of how this meristem is organized. You're seeing these green cells here are the center of where the stem cells are going to originate. And a lot of what we do is we cut this thing up and then we watch it come back, because of course plants are very, very good at regeneration. And we want to know about the structure of the root, how it comes back, right? I mean, if you want to know how something is built, you destroy it or you take it apart, and then if you can actually watch it reassemble, you learn a lot about how it's organized. So that's the main part of what we do. But here, especially in the Botanical Garden, I wanted to talk about some of our other work where we're looking more at the comparative systems of roots, how roots have changed over the course of evolution and how this drill has evolved in a sense. And then also some new comparative systems that we can use, including using new technologies like single-cell RNA-seq, to start to actually explore new species that we couldn't get to when we needed to invest years and years in markers and tools. Now with these new technologies, we can get at them very quickly. And I'll touch on that second. So let me tell you first a story that's just coming out, about our work on some of the original roots in evolution in land plants. And this is going to be in the lycophytes. But first, let's talk about what a root is, right, at the cellular level. So you all know, right, xylem, phloem, pericycle. This is the radial organization of the root. And remarkably, as far back as you can go, where there are true roots, and we'll take a look at that in a minute, you see this arrangement. Even the endodermis, which is the layer that's protecting the vasculature, that's actually going to form sort of a dry wall, a barrier that will regulate things in and out of the root, a cortex and epidermis. But of course that's the vasculature. Then there's almost always some kind of stem cell, and a root by definition this will become important in a minute, has to have a cap, right? That is, there are cells that are down here, there are cells that are up here that are giving rise to all these files, right? That's where all the growth of the root comes from. But then there are cells down here that are giving rise to daughter cells in the opposite direction, downward. And of course, those cells slough off, mostly, they're shed from the root, and that's why it's short. But this cap is an important part of what makes a root. The other defining feature is that it turns toward gravity, although this is variable, right? You know, roots can go sideways too. And it has root hairs. Okay. So that's the system that we're working with, is just an overview and a reminder. And when it comes to root evolution, the ongoing theories is that despite the fact that you see roots from here out very early, of course moss doesn't have even a true vasculature, but you evolve a true vasculature somewhere in here, and all these large-scale, right, taxa have roots. But the ongoing theory is that roots actually evolved a second time here, once here, and these roots do not have the same origin as the roots we see in quote-unquote higher plant, flowering plants and seed plants. So we're going to focus in here at the, what appears to be some of the earliest roots, and they have all those features that I talked about. And that's from this genus Selaginella is what we use, and this has this very interesting, weird structure. And so here you should see a shoot, and the embryonic root is actually really small and it kind of dies out very quickly. But the majority of the root system comes from the shoots. But before it becomes a root, it becomes this thing called a rhizophore. So here you see these structures growing out. There's actually, when these fronds, which bifurcate in the shoot, bifurcate like that. There's two meristems on the dorsal side and then on the ventral side. And they will one of them will give rise to this structure here. And then you see this bifurcation. Those glycophytes are not necessarily giving lateral appendages, but rather they're bifurcating their meristems. It's a very common feature of these basal plants. But there's been a debate for a very long time that out of this dorsal, or this angle meristem, where these two meristems grow out, there's this thing called a rhizophore. And this thing lacks a true root cap, and it lacks root hairs at first. And then later on, that structure will bifurcate, and then by this time, it's a true root because it'll develop those root hairs. So it's exactly 150 years ago where this structure was described, and there's debate over what it really is, right? And I'll get to the significance of the debate in thinking about the evolution of roots in the earth. So here you see again, and I have to acknowledge here that it's two different species of the Selaginella, but these are where the nice micrographs came from. But here you see one of the big differences in the rhizophore, where in lower plants, right, instead of having all those initials like the first picture that I showed you of Arabidopsis, where you have all these initials ringing the quiescent center, it's almost like the QC, the quiescent center, and the initials are all bundled into one cell, especially in basal plants. And it's this cell, which is going to divide off of many faces, that's going to give rise to all the cells of the root. And that, in the rhizophore, is right at the tip here. You can see that triangular cell. In the shoot, it has the same arrangement, where it's at the surface that this apical cell exists and gives rise to all the other cells. So the stem cell all, totipotent stem cell all in one cell. Now, when it becomes a root, the anatomy changes very dramatically. And here you see that cell, the apical cell, or the stem cell, initial cell, marked here, actually moves internally. It doesn't move. It changes which cell is the initial, becomes an internal cell. And then you have a true root cap and this thing called a calyptra. But now it's looking exactly like a root. It has all the features of the root, it's going to develop root hairs, it has the cap, and it's going to move into the soil. And I described this, right, where you have this branching, you have this dorsal meristem, and that almost always gives rise to another shoot, say here, and then this ventral meristem gives rise to this rhizophore. So that's going to become important in a minute when we start thinking about the issues that surround the identity of this organ. Okay, so before I do that, there's this classic experiment, which some of you may recognize, right? From what, 1957, where you can take callus and you can take an explant from a plant and treat it with auxin, and it becomes callus. This is like a very fundamental, undifferentiated plant cell, totipotent, pluripotent. And then if you treat with a different concentration of either auxin, high auxin, low cytokinin, you can make roots. If you treat with high cytokinin, low auxin, you can make shoots. So there's this bifurcation, there's this ability of plant cells in the treatment to actually have this difference in fate if you can bring cells back to a point, and this is brought back rather artificially. But the issue, let me skip over to here, and we'll go back to there. The issue in the Selaginella and all the related species in these early land plants is that this angle meristem seems to have that same plasticity. That is, it can move back and forth between root and shoot, and I'll show you that in a second. In addition, right, the rhizophore had all these shoot properties. It didn't have a root cap, had the apical cell, yet it's going to eventually give rise to a root. So there's this 150-year-old debate: is it a root, is it a shoot, or is it some unique organ? And the significance of this debate is that perhaps this represents this period in the evolution of roots where you have this organ that can kind of go either way, for lack of a better word. And so you have plasticity, you have this organ that's plastic first, and then eventually there's a kind of an assimilation of those two developmental programs into a fixed root and shoot. And so does this represent this early plastic organ? And so is it a root? So that's not a debate then. This is just an early stage of a root. Is it a shoot that then can sort of transdifferentiate into something else? Or is this a unique organ? Okay, so for one, what you can show is that there is this plasticity. And so this is a pretty complicated graph, but what you're seeing here is the dorsal meristem. And the thing to notice is that if you treat it with auxin, you can see that many of them will convert to the ventral meristem. That is, a rhizophore that will eventually become a root. And vice versa, if you take the ventral meristem on the bottom, which is normally going to become a rhizophore and a root, and you treat it with auxin inhibition chemicals, you can switch its identity to a shoot. And the key, this is an old experiment, and it's very well known. So you see how this organ has this dual ability to go either way, to switch its fate. So again, that may represent this very early stage in the evolution of a root, where you just have a generic organ, a blank organ, that can then take on these two fates. The other key part of this graph, and this was something that was unique to our work, is that we can now map to about, these are days after the emergence of that meristem, how many days after it's still plastic. So the idea, I'll go back to here, is that we're going to sample, we're going to ask this question rather than just still the anatomy is important and the growth is important, but we're going to start asking this question now using sort of all the information we can get from the mRNA, that is RNA-seq, and sort of try to label this, identify this organ based on these thousands and thousands of markers that we can sample. So the idea is we take the rhizophore at this point where it's clearly a rhizophore, it's no longer just an early meristem. It has not developed a root hair, it hasn't developed a root cap or root hairs. It does grow downward, it has no pigment, but it's still in this window where it can be plastic. So that's very important. Then we take the dorsal meristem, that shoot meristem, at exactly the same time. So we've given every opportunity for these to resemble each other. And then we take the mature root, say at that point. And we microdissect the meristem, that is, we just take the meristem, and we are going to generate the RNA-seq patterns, and then we're going to look at those and compare it. Okay, so what you see here is the classic heat map, where you see that the genes are on this axis, of course, and then the samples are on this axis. This is another way of looking at the data, where you're mapping in 2 dimensions the overall dissimilarity, right? This is MDS, which stands for, what does it stand for? Anyway, it's like the principal components analysis, multi-dimensional scaling. And what you're doing is separating them based on their overall expression. So interestingly here, what you see is that the rhizophore is very much separated from the shoot and the leaf. Okay, that makes sense, it's kind of going to grow down and become a root, but it's also equally distant from the root. And when you look at the expression pattern, yes, it does share a little bit of expression pattern with the root. The yellow are the genes that are upregulated, and so there's some sharing. And when you look at this overall hierarchical tree, you see that it's most correlated with the root, but it's kind of poorly correlated. And here's just sort of a control, leaf and shoot, and they're much more highly correlated. And then you can look at all the overlapping genes, and you can see that the rhizophore is quite distinct. It has a number of transcripts that are unique to the rhizophore. And then you can look at unique markers, and so this is really interesting to look at the genes that are specific to the rhizophore. But basically what all this is showing you is that actually the rhizophore does seem to be this unique organ that has arisen in evolution. We don't seem to have another parallel for it. It doesn't resemble its own root, and it doesn't really resemble its own shoot. So that, we feel like, comes a long way toward answering the question. We can look at some of the characteristics and look at the genes as they've been annotated in Arabidopsis, and they're orthologous, or at least homologous, to the Arabidopsis genes. And we can see that rhizophore indeed has this sort of allegiance to the shoot, where it sort of does have some photosynthetic genes, even though it's not green. So it does sort of bear some resemblance of the shoot where it originated. But then it has a lot of resemblance to the root. So here are some shared root characteristics, including cytokinin. So we didn't find any unique genes that were, any unique auxin signal transduction genes that were unique to the rhizophore or the root, or the rhizophore and the root. But we do see some cytokinin responses, and these can shape the auxin responses. So these may make some of the affinity or some of the uniqueness of the rhizophore. That we took a little more look at, and we took the auxin response factor. We don't have transformation, it's kind of a limitation of this model. We can't actually test some of the molecular hypotheses, but we can transiently transform the cells. It's hard to see here, but here are the untransformed cells. And what this is an auxin reporter that when it's reporting the presence of auxin, it's going to light up in GFP here. So you see this dramatic difference, but what this promoter represents are the binding sites of the auxin signal transduction system. So it really shows that auxin signaling is working in Selaginella to presumably make a rhizophore and a root. So this is kind of an anomaly, we think. There's been more discussion. There's a recent paper by Liam Dolan, actually on a fossil finding, that shows that the rhizophore, well, didn't call it a rhizophore, but there's an early structure at the base of the lipophytes that actually resembles a rudimentary root without a cap, without root hairs. And so it's believed that that's a precursor to the root. That's what they argued in the paper. So that is proof in the paper, at least, that roots probably arose separately in the lipophytes than they did in the seed plants and the ferns. Yet, there is this common signal that initiates both. So the ongoing model, whoops, the model is here. The ongoing model is that, yes, Selaginella, there's a fuzzy kind of notion of what makes a novel structure, right? The root may have been invented twice, maybe a cap was invented twice, but what's made the opposing signal to the shoot, a root, is actually quite common, this auxin signal. So I think we have to sort of get to the bottom of this, is what the role of auxin is. And so here our model is that the rhizophore is induced, and we have some separate data that shows that the rhizophore, when you treat those dorsal meristems with auxin and they become roots, they go through a rhizophore stage. And there's some characteristics we can tell. So it doesn't seem as though only the root is induced by auxin, but both the rhizophore and the root. And then we're not sure if auxin separately is needed to move from the rhizophore down to the root. Of course, in the flowering plants, auxin, it's simple. Auxin plus a little bit of cytokinin make a root. It seems that in the early plant plants, auxin does make this opposing meristem, and rather than maybe it is a unique organ, but it seems to be more of a unique stage that's been lost in higher plants. That is, this rhizophore is an intermediate between the angled meristem and the root, and that to become a root, you have to go through this stage, although that stage is pretty unique. So you feel like this sort of tells us that this is quite a unique meristematic structure that sort of was invented and maybe lost in somewhere in evolution. So actually this paper is just accepted, so we're going to get that in, so it's very timely, unpublished. And now I want to, if you guys have any questions, that's good. I think we have time. Let me check where I am. I'm going to try to end on time. I'm going to switch to a news story. So if there's a question that's fresh in your mind, I'd be glad to take it, but I'm also happy to take it at the end. You can hold it. Yeah?
Do you have some fossil evidence for these lycophytes and the seed plants that would suggest something that what would have been the ancestral state?
What would have been? Yeah, this is pretty much the paper that. So all the fossil evidence, the reason up until recently why there were thought to be two separate origins was because the fossil evidence at the base, remember I showed that very simple tree, at the base of the lycophytes, all the fossils they had lacked roots. This, roots are not always that well preserved in the fossil record, but that was the evidence. But then this paper by Dolan, they claim to have found one of these basal species, and they actually did sections of the fossilized rock. And so they got sort of very nice cellular structure, and they show that there is this organ that's not the shoot, and they go to great lengths to show that it's not the shoot, that's growing out, that's missing all the features of the root, but very well could be the early root. So that's the evidence so far. There's no other evidence in reality. So it's all pretty thin. Yeah?
I was wondering if the fact that in lepidodendrits, fossil lycophytes that lived in the Devonian,
okay,
they were huge trees and they had organs that branched at the base. And they had something that I'm going to call roots, but I'm not sure if those are roots, because they had a phyllotaxis.
They had a phyllotaxis on the shoot.
Yeah, I mean these organs that serve for anchoring and for absorption came out with a definitive phyllotaxis.
Right, so they more resemble a shoot.
So they're thinking they're more homologs to leaves, maybe.
Right.
Which are not the leaves that you see in seed plants and angiosperms.
Right. No, for sure, they're a different set of
That's
leaves.
a completely different
But are you're wondering how those trees anchored themselves, if not with roots?
No, my question was if those things that came out with a definitive phyllotaxis have something to do with this story.
Oh, I see. I don't know. I pay much less attention to the shoot than I do the root. But I am not sure where phyllotaxy emerges. One would argue if you see the phyllotaxy at the base of the lycophytes, that predates the root, and so that it would be coordinated with the origin, say, of the rhizophore. the phyllotaxy, right, everything is bifurcating in this species.
Right,
But the leaves, you're saying, may have some phyllotaxy to it.
Yeah.
And I haven't paid
it was
attention.
something spiral.
Right,
So you have bifurcating organs, like the main shoot,
yeah,
going into the branches in the top, but also into the anchoring organs
right,
at
that's
the bottom.
bizarre, isn't it?
And then on those, you have organs coming out with a spiral phyllotaxis.
Right. So that would argue, and it's part of the argument, I think I see what you're saying. It's part of the argument of the rhizophore that really all you did was take a shoot and kind of turn it around and make an anchoring organ. It hadn't really evolved into a full root yet. And that the rhizophore is some kind of intermediate. And the debate was, well, maybe it was still just a shoot that was heading downward. And then now, all of a sudden, you develop the ability to develop specialized root features out of that.
Yeah.
Anyway, we can talk about that more at that break. Yeah.
Quick question, like does the structure of the root in lycophytes, how does it compare to the structure, like in detail anatomy with the other,
With seed plant.
seed plants and ferns?
Yeah.
I mean, could you propose that they are homologous just based on the anatomy?
Based on the anatomy, I would want to call them homologous. It's quite remarkable how many features they share, right? Besides the vasculature, which you could say is trivial, that's all over the plant. The cap is shared by both, and it's a quite distinctive feature, and it contains amyloplasts, right, to help point the root toward gravity in both cases. There are root hairs in both cases. There's an endodermis, which you can stain for suberin in both cases. So it has all the features. If there's any way to define all the roots and in the angiosperms or in the seed plants, those are the features, and there's nothing else. And then everything else varies. And Selaginella has them all. What does differ is the structure of the stem cell niche, as we would call it, right? Where you have a single cell, but that's very characteristic of basal plants, is it's a single cell that's giving rise to all the cell types. By the time you get past fern into pine and say then the flowering plants, you have these much more complex stem cell niches. And they don't return and they don't have these single cells to give rise to an entire shoot apical or root apical meristem. So that is a distinguishing feature, but that's a distinguishing feature not just of roots in basal plants, but of all meristems in basal plants. So, and then the more we look to draw on our work in regeneration, the origin of cells doesn't really matter. The plant can produce, we're finding in regeneration, the plant doesn't really produce new cells from the stem cell niche right away, but the patterning just takes care of it and makes all the right cell types, the cap, the endodermis, etc. So how much the meristem organization matters, I don't think it's that much in making an organ. So yes, anatomically, they share all the features. Yet, the evidence is more and more pointing that these are separate origins. Quite a convergence. Yeah.
I want to understand the rhizophore, so it lacks a root cap. Right?
Yeah.
How about the tropic responses? Are they gravitropic, allotropic? Are they positive, negative?
Yeah, they're
So are they
gravitropic.
more like a root or like a shoot?
In terms of gravity sensing, it's more like a root. So it does have that feature. In terms of meristem organization, it's more like a shoot. In terms of gravitropism, it's more like a root. In terms of pigment, it doesn't have chlorophyll. So it's more like a root. So that was where all this debating for 150 years came from. They had, they had anatomy and they did a nice job, but it was stuck somewhere in the middle. But it shares these features. I guess gravitropism is important, but maybe, as we were just discussing, you can just make a shoot gravitropic and turn it around. It's not necessarily a distinguishing feature, the distinguishing feature of a root. So, yeah, they share a lot of features of both root and shoot, which was what made it hard. But by and large, when you look at the transcriptome and you look at the function, it really looks like a very separate stage. Okay, I'll move on, and then you can always come back and ask more questions. And now I'm going to switch topics, although we'll stay on the same idea of what makes a root and how you put it together, and differences in roots, but we're going to switch to angiosperms. And here, this is a project that's getting underway. We're sort of about halfway through it, and I'll show you our progress, also unpublished. And it's by postdoc Carlos Ortiz Ramirez, and he's done a great job in the lab of developing this whole system. So of course, here I'm showing you another big difference in roots, maybe now more subtle. Here we have Arabidopsis and maize. You can get a feel just from these sections that Arabidopsis, of course, has this very, very simple structure where all of the cell types that we looked at, and that was the diagram that we looked at in the beginning, all the cell types come in one layer, right? It's a very simple root. Maize is much different structure, right? It has a much bigger quiescent center, which would encompass maybe 1,000 cells down here. Very, very importantly, it has multiple layers of cortex. And cortex layers, of course, are really important. One of the major roles of the cortex is to host mycorrhizal symbiosis, right, with fungi, and they're going to invade the root, and they're going to branch out in the inner cortex. So this specialization of the cortex and the elaboration of body plan here in the, or patterning here in maize is very important. And of course, there's about 150, million years between these. So we're really interested in how cell types evolve and how they change. And there's very analogous cell types here. And we're interested in how that whole patterning system changes. So this was a tough problem up until a short while ago. Basically, it took us many, many years to develop all the markers for the cell types in Arabidopsis. I'll show you how we can actually sort the cells with a fluorescence-activated cell sorter and get profiles of the cells, but it was tough. And so Carlos is going to be using some new techniques to try to quickly map all the transcripts and find out what's unique, just like we did find out what was unique to the rhizophore. Here we're doing it on a finer scale. We want to know what's unique to specific cell types. So that revolution comes from all these techniques, which I'm sure you've heard of. We had used in our original paper, like it's not even 4 years ago, 2 years ago, it came out. These techniques where you can, you can apply the leaf cutter and the fungal enzymes that will break apart the cell walls and make protoplasts. We did it in fluorescent lines, and then we put each cell in a well of a 96 well plate, and you can make RNA and just miniaturize everything. Now, like a year after that technique came out and increased the scale, there are these techniques where you can put the cells in these microfluidic devices, and you run the cell in one channel. You run a little bead, I'm sure you've heard of this, that has RNA, that will capture the RNA. So RNA is poly A, the bead is poly T. I think I have that down here. Right? And this is how you make cDNA. That's the standard technique that's existed for decades. But on that little bead, you could put a DNA sequence that serves as a barcode. And every little, every little primer on that bead has that same barcode. And then this bead has a different barcode right there. So then you can process these cells. You can do the early steps in these little droplets that you're forming, right? The cell meets the barcode, then they are together in a little droplet, then a bead of oil, or a stream of oil comes and makes separate droplets. And I thought this was the coolest thing when I saw these microfluidic guys using these technique. Now it's like 5 or 6 years ago. And so then you can run thousands and thousands of cells through this, and then you can do the RNA sequence right at later steps. You can purify the beads and then process everything together, because all the transcripts have this barcode, so you can map them back to the cells. So you get to collect all this information. So this has been commercialized. This is the dropseq version. It's been commercialized in an instrument called 10X. I'm not sure if you guys are using that, but this is the technology we're using. In the last year, there's been another technology where you can increase the throughput now to hundreds of thousands of cells in a single experiment. And it's basically by using sort of combinations of different barcodes so that you uniquely mark a cell with 1 in 50 million barcodes. And that's increasing the scale even more. So this is kind of like the moon race of molecular biology. People are increasing the scale by an order of magnitude. It's been about every year and a half or so. We're at this stage. We're at the orbiting the Earth here, so we have, the moon is coming. Those techniques are, still in development. So Carlos is going to use these techniques, and the idea is that now, rather than try to collect single cell types, it took a long, long time to make those markers in Arabidopsis. Maze is even harder. The regulatory sequences are longer. You're often not capturing the whole thing. The screening is harder. It's a thicker root. So now the idea is you can just sort of cut off the meristem and you can generate, each one of these is a single cell. This is a principal component like plot, just like we looked at before with the rhizophore and the meristems. That's basically mapping the distance between each of the cells based on their total transcriptome, right? So it's dimensionality, reduction, and you're looking at clusters of cells. So you've just cut this off and you've looked at all the cells anonymously. And of course you've lost all the context of where the cells come from. So that's a problem. So, but you can see on the plot here that you do get unique clusters of cells. And that's the idea behind this technique. Any single cell is very, very noisy. You're missing, there's a lot of false negatives. You're missing data just because of the nature of preparing the cDNA at the level of a single cell, right? But if you see these cells all grouping together, you know they have a common identity, or you surmise they have a common identity. Then you can look for the transcripts that are in every single cell, but that are much more common here than they are to here. And you know then some of the transcripts, you can start to map all the transcripts that are unique to those cells. Okay, but the problem here is we're kind of blind, right? We've isolated all these cells. We have a few markers for maze that we know where they belong, but of course that's very noisy, so we can't just use a few markers. So we're kind of in this situation where we have a lot of puzzle pieces and we don't know how to put them together, right? So what do you do when you have a puzzle? This is a 10,000 piece puzzle, and you need to put it together, and you want some help, right? You look at the box, right? So we have a box in other species, in Arabidopsis, right? When I was telling you about the markers, we have each single cell that's meant to represent a fluorescent line, where we've taken those cells, we've done the RNA-seq on the whole group of cells, and we found, this represents genes are here, so maybe 100 genes that are quite unique to the ground cell, that's the lateral root cap, okay? And so we have a great map that we could use in Arabidopsis. The trouble is we can find homologs, or, if we're really being bold, we can call them orthologs from Arabidopsis to maze, and maybe that'll be conserved, and we can use the Arabidopsis to map the maze cells, to find out which the maze cells. But it doesn't work. There's been too much, and this gets at that question of what is homology. It has all the same cell types, but the transcripts in general don't map to the same cell types. So the evolution seems to have switched around the cell type quite dramatically. You can do it with paralogs, but we quickly lose a signal. And there's certainly some things that are conserved, and I'll show you there's a lot of things that are not. But we quickly lose the signal. And after all, this is quite a divide between the monocot and dicot. Okay, so Carlos was stuck, but we did notice that here we had stains. Okay, the animal people have all these stains where you can stain for nuclei, or you can stain for lipid bodies and things like that. And they're supposed to stain every cell and identify subcellular organelles, right? But they're no good. They stink because they don't stain every cell. They're very variably penetrable. But we noticed this variable penetrance after a while, and it's actually remarkably consistent from species to species of root at least. And we have that nice arrangement in root, right, where, we always know the outer layer is the epidermis, then comes cortical layers, and then comes extra layers, right? So from the outside in these dyes, you could capture kind of a fairly unique set of cell types, right? They're not all mixed in, they come in these layers. So Carlos realized that if you took the ratio in the FACS, and this is a FACS plot, here's a cell, so that, X and Y axis may represent, or does represent, the ratio between the blue dye here and the green dye here, and using those ratios, you can map cells to, sorry, let me stay there. Sorry for all the back and forth. You can map cells to this position, this position, this position, this position. And he did that, and the FACS is a great instrument. You can look at 1,000 cells per second, 10,000 cells per second. You can grab them, put them on a microscope, and he measures the ratio in fluorescence, and lo and behold, they do come from the different regions of the root that he can predict. So in a sense, he's made the box, right? He's made the puzzle box. He's got an ability now to gather the epiderm, what is it? P7, no, this is the steel, the inner cells, then the next outer cells, then the next outer cells, then the epidermal cells. I believe that's right. Yeah, I said it here. So now we can map the cells, right? And now we can, in theory, do this for any species really quickly. We don't need to develop markers. So we can get this true comparative cell type expression profile of the entire transcriptome. And again, like I showed you before, in the Arabidopsis, we can now map these markers to different regions of the root. It's not quite that precise, right? We can't get down to the single layer, but we can get down to sort of epidermis, cortex, steel, so we can do this rough mapping. And we use some markers that we know. There have been in situ hybridizations, and we can we can check ourselves, and we can see that we're usually quite on the mark, that the cells that represent this profile came from the right region. Okay, so we're good. We have markers now. And now I should put more of the technique here, but we use this method. So now, okay, we have the box, we have the cells, and now we have to look at the cells, puzzle pieces, and figure out where they are in the box. That's my analogy, but we have to do that with transcriptional data. So what we've done here are develop a set of techniques where we can let the markers, because any given marker, remember, this is noisy data. The single cell systems, you can't rely on any one marker. You have to use as many markers as you possibly can, and here we try to use 10, 100, more like 100. So what you can do is you can just look at the cell, and you can rank. Do I have it here? No, I left it on the note. You can rank the number of reads you get for each transcript. So the most highly expressed genes to the most lowly expressed genes in the cell. Then you can take the markers that we've identified from the sorting and the ratios, right? And we could say, does that cell, are the most highly expressed genes, this is the intuitive way, are the most highly expressed genes come from the epidermis, right? Then you would tend intuitively to say, if the most highly expressed genes are the markers that are unique to the epidermis, this must be an epidermal cell. It's a simple idea. It's a voting technique, and we do some scrambling to get some statistics out of it. And then in our regeneration work, we can even tell if a cell has a chimeric identity, has two identities. So it's pretty simple, but it's very powerful. And so now we can take the whole clusters, and we could do this voting, test their identity, and we could come up with an identity for each of the cells in the cluster. And you can see here, I think that says endodermis. All these cells turned out to be endodermis, right? So now we're not as lost as we used to be. And let me make sure, I'm going to go over. Anyway, long story short, now we can map, we have some markers like the endodermis where we can tell the exact layer. This should be this is epidermal markers. So we can identify each of these clusters. And we did that, and now even you can see, you can start to, go back here, you can start to see that even though we could not distinguish the separate files of the cortex, we can see clusters between the steel and the outer cells that map to cortex that actually look like they represent the different layers. So we can increase the resolution with the single cell. So this is kind of fun. Now we can look at the whole genome and we can look at things that evolve. And one story that came out of this in the last few minutes of this, and I won't take too much time on this, is that we noticed some anomalies. We said right at the beginning that it was very hard to map transcripts from Arabidopsis to maize, and we saw that this classic gene, this is short root, in Arabidopsis, it's expressed in the stele, and it's one of these really important organizing genes. It communicates, it actually moves through the plasmodesmata into the cell next to it. It creates this endodermal identity. It allows that cell to divide, and it creates a pattern. And that's a classic role in one of the early pieces of evidence that transcription factors could move. Here, instead of seeing it in the middle, we see it in the cell that it's supposed to move to in maize. And the single cells led us to that, and here we made the marker, and lo and behold, the marker backs that up. So again, that model is in Arabidopsis. Here's the transcriptional fusion, here's the protein. You see the difference. The protein moves over one cell, it induces this other gene, which leads to the division. So you have this kind of, and then ultimately, these two together will signal back, and they'll pattern the vasculature. So really beautiful, elegant system. In maize, here you see the short root gene. It's expressed in that outer layer where it's supposed to move, and then it moves even further. So the hypothesis is that it can create these divisions. So what it's doing is creating these extracortical layers. And it looks like that's what's happening in maize. So interesting difference. The other really interesting thing is here's that other gene, scarecrow, which just makes the division, and it's expressed in the endodermis. It stays where it is. If you look at the difference between the transcript, the mRNA, and the protein, they're in exactly the same cells. But in maize, that scarecrow gene, here's the difference between the transcript and the protein. It actually moves in the opposite direction, inward. So while here's a sort of something that looks like it's coming out of the single cell data, we've sort of reconstructed this important, or we've seen a major evolutionary difference in the way the root is organized, in the sense that in Arabidopsis, I'm using the laser, in Arabidopsis you move from the yellow cell to the green cell, then back to the yellow cell. Here, and so it looks like the yellow cell is kind of the organizer. Here, it looks like the green cell, the endodermis, is the organizer, because it's the origin for signals that move outward and inward, and it definitely, through the scarecrow mutant in maize, it patterns that inner cell types, the vasculature. So very important change, and again, we're sort of pursuing this. We think it has connections to Krantz anatomy, but we can talk about that. And so this is sort of an ongoing story, but shows you the power, I think, of being able to dissect the organ at the all at once at the single cell level. Okay, so we'll skip that. Let me just credit people. I didn't talk about John. He just got his position. He did all our regeneration, recent regeneration work. Ramin makes those beautiful movies. Carlos is totally responsible for that last part of the single cell work in maize. I didn't talk about Marcella's project, but that's another regeneration project where we're looking at the chromatin and changes in the chromatin. And I'll stop there and let you guys ask any questions on that last part if you have any. Thanks for your patience.
¿Preguntas?
Thank you for this very interesting talk. The difference in Arabidopsis and maize in terms of the endodermis producing the transcription factor and the subsequent. So this shift, is it, I'm not sure, is it like a monocot-dicot thing, or is it like, because there's multiple cortical layers that this, what, like I'm trying to understand evolutionarily what sort of a shift is this?
Right, that's a good question. Like, what's the significance in terms of the evolution of features? And I think you hit it, that maybe we'll go back to one, because I think you raised a good, there's two movements, right? There's two changes in the movement. One is that the short root is moving in this direction much further than the Arabidopsis does. And so what the working hypothesis, which seems very good, is that this is a monocot-dicot split, or at least a split where you have a very simple arrangement with one or very few cortical layers to making multiple cortical layers, like a good maize root would have. When you need to make those multiple cortical layers, you want short root to move further, make more divisions. So that would be an adaptation for that. But this inward split, people have looked at rice, and it does not look like in rice, which is C3, right? Not C4 like maize. It does not look like in rice that this scarecrow moves inward. There's been mutant analysis in scarecrow that shows that it has a much bigger role in maize in patterning the vasculature than short root does in Arabidopsis. It actually does have a role by helping other things move back. But scarecrow. But scarecrow has this role in what appears to be really changing the vasculature. For reasons that are hard to get into, for one, if you look at the difference between C3 and C4 anatomy, right, this is the photosynthetic evolution, there's major innovation in photosynthesis, the vascular strands are closer to each other. Of course, the bundle sheath is the place where a lot of the changes have happened. We think short root being in what is equivalent to the bundle sheath layer creates some changes to the bundle sheath photosynthetically that are a major advantage or a major innovation that is needed in C4. So putting short root, rephrase it and make it more clear, putting short root from the stele into the endodermis may help in the endodermis. The equivalent cell in the shoot of the endodermis is the bundle sheath. So putting it there may change the photosynthetic, and there's some evidence for this downstream of short root, may change the photosynthetic properties of the bundle sheath that enable C4. Okay, then the vascular strands need to be closer together in a C4 plant. And we think that the hypothesis is there's something about what scarecrow, the other one, is doing when it moves inward that changes the vasculature that enables this closer pattern. That's a little bit of hand waving, but it definitely has a major change. So what we're doing is now trying to take other C4 plants, like Ceteria, which is a nice model, millet, and we're trying to do the same, quickly make these maps and see if you see the same pattern. It's a great question. Yeah.
Do you mean with this relation in the root and the leaf that the vascular tissue is connected since the beginning of the development? So that's why the sign is going to the leaf?
Yeah, that's why what goes to the leaf? That's...
Yeah, from the root, if you have those expressions.
Oh, why I would go from
Well,
yeah, you raise a good point. We're looking at the root, and of course C4 is only important, or should be only important in the shoot. I don't know. Maybe once you start to make these changes and you need them for the shoot, you would be constrained to do the same thing in the root. It's a good question. Or maybe it happened in the root first, and that was a pre-adaptation to what's happening in the shoot. Or maybe there's aspects of C4 metabolism that are happening in the root, and the root is doing some of the work, although there'd have to be a lot of transport of metabolites. Yeah. But I can't answer. Yeah.
Hi, Ken. Is there only one gene of a short root in maize?
No, 3.
Okay, so maybe the other ones are moving differently?
Yeah, you know, of course, making the constructs is the longer, harder part. We have two of the constructs. Okay, so all three isoforms all three paralogs are expressed in the endoderms. So they've all changed their location. One of them is expressed very highly. We're trying to make the translational fusion there. We have a reporter for one, no, we don't have a reporter for another yet. So we have the transcriptional regulation for all three, and we're making the translationals to see the movement. And of course there's 3 scarecrows as well. Yeah. Yeah. Okay.
So I just wanted to point out about the homology thing that you mentioned when you were trying to see like if the patterns were similar. But I feel like just because the structures are homologous, that doesn't mean that you should expect the same transcriptome patterns. You know, like what homology says is that these structures were inherited from a common ancestor, but they have undergone transformation, and you know, especially in this case where a lot of time has passed between these ancestors, I mean, of course, you should expect some similarity, but not necessarily like the same patterns. I don't know.
That's the lesson we learned. But yeah, I mean, well, we had seen, say, from rice to Arabidopsis conservation in things like scarecrow, right? Here we see differences. So, you know, we have this idea in development, but we're very Arabidopsis centric, right? That's where the patterns were learned. We have this idea that the core regulators should be conserved. Maybe there's not enough of them to give a good signal, but no, I think you're right that what's constrained are the properties, right? The endodermis needs to be a layer with lignin and needs to get suberized. And of course there's enzymatic pathways that need to be there, right? But how they're regulated and the rest is, over this amount of time, is pretty non-constrained.
And also my understanding is that these transcriptomes are like kind of snapshots, right? And they depend, I mean, there's going to be variation depending on the exact, like the stage of development or, you know, the environmental conditions. I'm not sure, I don't have a lot of experience with these.
Yeah, we've done a bit of that, and depending on environment, you know, we've treated the roots and resorted the cells. There are changes, and plant cells are very plastic, right? They're responding to the environment dramatically because, you know, the plant sessile. But by and large, and we've used that to those different cell types and different conditions to find the unvariable genes. And there's plenty that really, depending, no matter what the condition, really defines the identity of the cell in a single species. So the range of changes in transcriptional development in evolution are not reflected so dramatically in environmental response in a single species. But they do vary. I mean, along those lines, it will be, you know, your whole point, and I think it's a good one, that there's all this change that underlies this conservation of features, right? That it'll be interesting once we start to move away, you know, we'll take maize, we've taken a real extreme from monocot to dicot. You know, once we start doing monocot to monocot and see the rate of those changes, it'll be really interesting. Yeah.
Are we sure that there's no rhizospore in dicots, in angiosperms?
Yeah, that's a good question. You know, tomato will send out these roots from the shoot system. My postdoc, my former postdoc, Idan, studies that. But I think those structures, the reasons why they're not called rhizophores is because they have a cap right away. They have a root cap, root hair.
But how many roots we know about plants?
I mean, in the good old days, right, where we focused on 1 or 2 plants, they were very good at looking at a variety of species and doing careful sectioning. You know, when I need to go back and look at the diversity of a structure, I go back to the 40s and 30s and before then. So I think it's I think it's been pretty well studied. I can't guarantee that there's no rhizophore anywhere.
Bueno, si ya no hay más preguntas, los invito a pasar al café y galletas y a platicar más con el doctor.